Okay, so welcome to this next video in which we are discussing heart failure and heart failure medications. Okay, right, so what we've just looked at is normal cardiomyocyte calcium signaling. Okay, so we've seen how in order to commence the contraction of the cardiomyocyte, what you need to have is a calcium spike. Okay, so cytoplasmic calcium concentration, or myoplasmic calcium concentration if you prefer, needs to rise from the normal level of 10 nanomolar up to around 1 micromolar. Okay, and we've discussed that the way that this occurs is that when the action potential propagates along the sarcolemma, what it's going to do is it's going to activate L-type voltage-gated calcium channels which are in the sarcolemma and which are within these dyadic junctions between the sarcolemma and the membrane of the sarcoplasmic reticulum. Okay, it's going to activate those L-type voltage-gated calcium channels to open. Okay, and they're going to allow calcium to move in from the extracellular fluid into the dyadic cleft, which is this portion of myoplasm in between the sarcolemma and the sarcoplasmic reticulum membrane. That calcium is then going to induce calcium-induced calcium release from the type 2 ranitidine receptors, and the type 2 ranitidine receptors will release calcium from the lumen of the sarcoplasmic reticulum, and they'll release a lot more calcium into the myoplasm than is coming in through the L-type voltage-gated calcium channels. Okay, and this release of calcium from type 2 ranitidine receptors is known as a calcium spark. Okay, and you'll be having this occurring in all of these different dyadic junctions all over the cardiomyocyte, and the addition of all of the calcium sparks together, the combined effect of that is going to raise myoplasmic calcium concentration up to form this spike, and then that's going to trigger the contraction of the cardiomyocyte. Okay, right. Then, to relax back down, what will happen is when the calcium gets high enough in the dyadic cleft, it will inactivate the L-type voltage-gated calcium channels and the type 2 ranitidine receptors. They will now shut, which means that we're getting no more entry of calcium into the myoplasm from the extracellular fluid, and neither are we getting calcium released from the sarcoplasmic reticulum lumen. Okay, now what remains is to remove the calcium from the cytoplasm, and it's the normal equilibrium mechanisms that are going to return you back down to an equilibrium. Okay, so SERCA2A on the membrane of the sarcoplasmic reticulum is then going to return calcium into the sarcoplasmic reticulum lumen. It will return about 75% of that excess calcium that's in the cytoplasm back into the sarcoplasmic reticulum lumen. Okay, and then the other major mechanism is the sodium-calcium exchanger 1, which is going to chuck about 25% of the excess calcium back out into the extracellular fluid, and then it will return you back down to this normal equilibrium where you have 10 nanomolar calcium concentration in the myoplasm. You have this equilibrium both at the sarcoplasmic reticulum membrane and at the sarcolemma membrane, where the amount of calcium leaking out of the sarcoplasmic reticulum lumen exactly matches the amount of calcium that the SERCA2As will be bringing back into the lumen at a myoplasmic calcium concentration of 10 nanomolar. Similarly, at the sarcolemma, the amount of calcium that's leaking into the myoplasm through some unknown leak mechanism will exactly match the amount of calcium that these two mechanisms, the PMCA and the NCX1, will extrude when the cytoplasmic calcium concentration is 10 nanomolar. Okay, right. And then we've discussed how sympathetic stimulation of the heart can increase the force of contraction of the cardiomyocytes. Okay, we've seen that adrenaline or noradrenaline will stimulate beta-1 receptors on the surface of the cardiomyocytes and hence activate protein kinase A in the normal GS cascade. Okay, the catalytic subunit of protein kinase A can then phosphorylate a protein known as phospholamban, or PLM for short, which is in the membrane of the sarcoplasmic reticulum. Okay, so in the sarcoplasmic reticulum membrane you have a lot of SERCA2As. Okay, some of those will actually be functional, others will be a reserve pool of SERCA2As that are at the moment bound to phospholamban proteins, and the phospholamban stops them from actually functioning. Okay, now the idea is that protein kinase A is going to recruit these reserve SERCA2As by phosphorylating the phospholambans, which are at the moment inactivating them, and when they phosphorylate the phospholambands break away from the SERCA2As, and therefore you liberate this reserve pool of SERCA2As, and they're now going to be functional. Okay, so you increase the number of functional SERCA2As in the sarcoplasmic reticulum membrane. What does that do? Well, it reshifts the equilibrium that we had here. Okay, so imagine that the cytoplasmic calcium concentration remains at 10 nanomolar. If we now increase the number of SERCA2As in the membrane of the sarcoplasmic reticulum, what's going to happen is they're now going to be pumping more calcium into the sarcoplasmic reticulum lumen. Okay, initially what will happen is the calcium concentration will still be the same, okay, and therefore the leak will still be the same. So now basically the amount you're moving in is no longer balanced with the leak, okay, out. So you're going to reshift the equilibrium basically. Calcium is going to build up inside the sarcoplasmic reticulum lumen. It's going to rise to a higher concentration until the leak is now big enough to completely balance the amount that you're bringing in. So as you raise the calcium concentration in the sarcoplasmic reticulum lumen, you will increase the leak, and you'll reform a new equilibrium, which will be the point where the amount of calcium you're moving out of the cytoplasm into the sarcoplasmic reticulum lumen with this greater number of SERCA2As in the sarcoplasmic reticulum membrane equals the amount of calcium that is leaking out, and that will be at a higher sarcoplasmic reticulum calcium concentration. Okay, right, so that means that because of this, your cardiomyocytes are now going to have more calcium stored in their sarcoplasmic reticulums. Okay, this means that when you activate the cardiomyocyte, okay, when an action potential fires along the sarcolemma of the cardiomyocyte, the amount of calcium that it's going to cause to be released from the sarcoplasmic reticulum is going to be greater because there's more calcium now in the sarcoplasmic reticulum, okay? And that means that the calcium spikes go up in amplitude. Basically, you increase the amplitude of the calcium spikes. And then when you have a greater amplitude calcium spike, that's going to recruit more cross-bridge cycling and therefore a greater force of contraction. Okay, so an understanding of that normal physiology is very, very important, okay, when we now want to consider what has happened in these hypertrophied cardiomyocytes. Okay, right, so we're going to look at certain things that go wrong with the calcium signaling mechanisms in hypertrophied cardiomyocytes. Now, unfortunately, I can't really, in most cases, tell you why these mechanisms have gone wrong in the hypertrophied cardiomyocytes. Okay, what we know is that the cardiomyocytes try to undergo this adaptive response where they grow to try and increase their strength, but somehow they don't get it right, and they end up destroying their calcium signaling mechanisms. Okay, so their calcium signaling mechanisms get hugely disturbed by the growth of the cardiomyocytes. Okay, and that's what then leads to these cardiomyocytes hugely losing their ability to contract, basically. Okay, so let's now have a look at what goes wrong in these cardiomyocytes. Okay, so the utter principal change that has occurred in the cardiomyocytes is that the amount of calcium in the sarcoplasmic reticulum stores, okay, goes down. Okay, so the amount of calcium in the sarcoplasmic reticulum at that equilibrium is decreased. Okay, and this effectively does the opposite to what sympathetic stimulation should do if sympathetic stimulation is given acutely, okay, which is that it should raise calcium concentration in the sarcoplasmic reticulum lumen and therefore increase the amplitude of the calcium spikes. In the hypertrophied cardiomyocytes, we find that the amount of calcium stored in the sarcoplasmic reticulum at this equilibrium is reduced. Okay, that's the most marked feature. Okay, and that means that when an action potential fires along the sarcolemma, the size of the calcium spike that that's going to induce is going to be smaller. Okay, so what we now want to look at is why is the calcium level within the sarcoplasmic reticulum reduced? Okay, well, there's three contributors to this that I'm going to tell you about. Okay, so the first one is that SERCA2A proteins are found to be reduced in the membrane of the sarcoplasmic reticulum in these hypertrophied cardiomyocytes. Okay, so SERCA2A is found to be reduced, and related to that, okay, making this situation even worse, phospholamban is found to be increased. Now let's think about what this is going to do. Okay, so let's draw a little picture here. So maybe this is what our healthy cardiomyocytes should be. We've got a bunch of circa 2As here that are not bound to phospholamban and are therefore functional, and then we've got some circa 2As which are bound to phospholamban and therefore are not currently functional but form that reserve pool of circa 2As that can be recruited into action by sympathetic stimulation. Okay, so this is what you might have in a healthy cardiomyocyte. The first thing that's going to happen in these hypertrophy cardiomyocytes is that the absolute number of circa 2As has reduced. Okay, so how can I do this? I'm just going to add one more circa 2A on here because otherwise it's a difficult number to work with. Okay, so let's put another one there. Okay, so what's going to happen, let's say we'll go down to 3 of them. Okay, so now we have less circa 2As in the sarcoplasmic reticulum membrane. So let's say we go down to 3 of them. In addition, we have more phospholamban, and therefore the fraction of circa 2As that are actually going to be bound to phospholamban is also going to increase. So we've decreased our absolute number of circa 2As, but we've also increased the amount of phospholamban, and therefore we've increased the fraction of the circa 2As that are going to have phospholamban bound to them. Okay, so now you can see that I've got 2/3 of the circa 2As here bound to phospholamban. Okay, and that's just a number that I've made up off my head. I don't know what the actual numbers are. Okay, this is just to get the point across here. Okay, so we can see that the 2 changes are that the absolute number of circa 2As has gone down from 4 to 3. Okay, again, that's a made up number just to get the point across. And because the number of phospholambans has gone up, the fraction of circa 2As that have got phospholamban bound to them has also gone up. Okay, so that means that both of these factors, both the absolute drop in circa 2As and the increase in the fraction of circa 2As that are bound to phospholamban, these are going to mean that the number of circa 2As that are actually functional, which are the ones that aren't bound to phospholamban, is hugely reduced. So it's gone down from 3 to 1. Okay, so both of these factors hugely decrease the functional circa 2As that are in the sarcoplasmic reticulum membrane. Now, of course, that explains perfectly why the calcium stores in the sarcoplasmic reticulum have gone down, because remember our equilibrium. Okay, this is such an important concept to understand. Okay, at equilibrium, the amount of calcium moving into the sarcoplasmic reticulum lumen via the circa 2As should equal the amount that's leaking out. Okay, we have suddenly reduced the number of functional circa 2As. Okay, what then would happen? Well, the leak initially would remain the same size because the calcium concentration in the sarcoplasmic reticulum hasn't had time to change yet, so you get more calcium leaking out than you would possibly be able to put in now. Okay, you'd shift the balance basically, so now calcium would net move out, and that would decrease the calcium concentration in the sarcoplasmic reticulum lumen. And it would decrease and decrease and decrease until the point that because the calcium concentration has dropped so much, the amount that's leaking out has now also dropped enough that it's equal to the amount that these reduced number of functional circa 2As is actually bringing in. So you'll reshift the equilibrium, and that new equilibrium will be forged at a lower calcium concentration in the sarcoplasmic reticulum lumen. Hence, how we have now reduced the amount of calcium in the sarcoplasmic reticulum at this equilibrium, and that then means that when you stimulate the cardiomyocyte, the size of the calcium spike that you get in the cytoplasm is going to be reduced, okay, and therefore the force of contraction is going to be hugely reduced. Okay, there is one other little mechanism that I want to talk about though. The circa 2A and phospholamban problem is most likely one of the most important things that happens. Okay, but also it's thought that there are problems with the type 2 ryanodine receptors as well. Okay, and to discuss this, I'm just going to talk a little bit more about the structure of a type 2 ryanodine receptor. Okay, so I'll just do this on this other page. So we're not going to go into the detail of type 2 ryanodine receptors in a huge amount. Okay, all that I want you to understand is that type 2 ryanodine receptors, they are these ion channels that are in the membrane of the sarcoplasmic reticulum. Okay, so here, this cylinder-shaped structure is going to represent our type 2 ryanodine receptor. and it's sitting within the phospholipid bilayer of the sarcoplasmic reticulum. Here is the pore going down the centre, and now the important thing to understand is that type 2 ranitidine receptors are not just made up of a single protein. Okay, instead they are made up of multiple proteins joined together. In fact, they are made up of 4 separate proteins joined together, so they are a tetramer. Okay, so it's not just one polypeptide, you have to make 4 separate polypeptides and join the things together. Okay, and each one of these separate polypeptides that makes up a quarter of the entire type 2 ranitidine receptor is known as a type 2 ranitidine receptor subunit. So I'll put RYR2 for type 2 ranitidine receptor and then subunit. And then the entire thing is called the type 2 ranitidine receptor. Okay, right. Now, it goes further because you can also attach additional proteins onto the type 2 ranitidine receptors. Okay, the tetramer of 4 type 2 ranitidine receptor subunits is the main part of the type 2 ranitidine receptor, but there are also proteins attached onto this tetramer structure. Okay, and each one of the separate type 2 ranitidine receptor subunits, okay, is going to have a little protein attached onto its cytoplasmic side, okay, known as calstabin 2. Okay, so I'll just try and draw this in. So these little boxes are supposed to represent little calstabin 2 proteins, okay, which you have attached to every single one of the subunits. So each one of the type 2 ranitidine receptor subunits has one of these calstabin 2 proteins attached to it, shown here in turquoise. Okay, right. So, let me just write its name out here. So its name is calstabin 2. Okay, but it also actually has another name, which you will see if you read the literature. Okay, so it's also called FKBP 12.6. Okay, now what's the origin of that name? Well, if you've ever studied the immunosuppressants, you will probably recognize FKBP. Okay, FKBP stands for FK506, which is a silly name for a drug. Okay, so before drugs are actually used clinically, when they're just experimental compounds, they're given code names like this rather than an actual name. Okay, so FK506 is just a code name for a drug. This drug now actually is used clinically and therefore has actually been given a proper name. Okay, it's now called tacrolimus. Okay, but many of the proteins that tacrolimus or FK506 binds to are named after the original code name. So FKBP stands for FK506, this drug, binding protein. Okay, so the BP is the binding protein, and then it's just the FK506 binding protein 12.6. Okay, so that's another catchy name for calstabin 2. Okay, now, why is this important? Well, I should probably say at this point that this theory is still a little bit controversial. Okay, there are bits of data which kind of oppose it. Okay, but it still is a theory that has some weight behind it as a model for what happens in hypertrophy cardiomyocytes. The knowledge about SERCA2a and phospholamban, that's much more solid. Most people agree on that. This little theory that I'm about to show you is a little bit more controversial. Okay, right. So basically the idea is that calstabin 2 molecules stabilize the type 2 ranitidine receptor in the closed state. Okay, so they stop it from opening until you get the calcium signal that causes it to open. So until you get that calcium sparklet in the dyadic cleft, and I'll just get my picture back again. Until you get the calcium sparklet coming through the L-type voltage-gated calcium channels, you don't want your type 2 ranitidine receptor to be opening. Okay, and the idea is that calstabin 2 is involved in making sure that the type 2 ranitidine receptor doesn't open until you get that calcium sparklet. Okay, right. Now, then what is thought to happen in cardiac hypertrophy is that the type 2 ranitidine receptor subunits get hyperphosphorylated by what is presumed to be protein kinase A. Okay, so what happens is the type 2 ranitidine receptors subunits get hyperphosphorylated. Okay, so I'll just draw one phosphate group attached to them, but I don't actually know how many phosphate groups they get added on. Okay, but they get phosphorylated at phosphorylation sites. Okay, right, and this is thought to be done by protein kinase A. So this is starting to potentially make sense, because remember, we're getting that chronic sympathetic stimulation, which is activating protein kinase A. So if protein kinase A is activated chronically, maybe it does cause the hyperphosphorylation of the type 2 ranitidine receptors, okay, which it wouldn't do maybe if it was only activated acutely. Okay, right, so the type 2 ranitidine receptor subunits then get hyperphosphorylated, and the problem with hyperphosphorylation is believed to be that it causes the calstabin 2 proteins to dissociate. Okay, so all of these calstabin 2 proteins, or the FKBP 12.6 proteins, they dissociate off, so hyperphosphorylated. Okay, right, and that causes the calstabin 2 proteins to dissociate away. Okay, now, what then happens once the calstabin 2 proteins have dissociated away? Well, now, the type 2 ranitidine receptor is going to open spontaneously, even with no calcium present in the dyadic cleft, or at least no high level of calcium present in the dyadic cleft. You might be wondering, well, the surely is the 10 nanomolar calcium that's present everywhere in the myoplasm. That's not sufficient to activate the type 2 ranitidine receptors, at least not normally, okay, not in normal physiology. Okay, so what now starts happening then is these type 2 ranitidine receptors start opening sporadically, basically, on their own, and therefore they start leaking calcium out of the sarcoplasmic reticulum lumen, into the myoplasm. Now, what effect does that have? Well, effectively, what this means is that you're going to increase the calcium leak, okay, because now these type 2 ranitidine receptors, they are contributing to this leak, okay, so now the leak is going to become greater, okay, that's also now going to help shift the equilibrium, okay, because, remember, we have an equilibrium where the calcium being moved in is equal to the amount leaking out. Okay, we've already shifted the equilibrium because we've reduced the amount coming in by reducing the number of functional circuit 2As. We are now increasing the leak. Okay, so what that's going to do is it's now going to shift the balance again. You're going to get more calcium leaking out than you're bringing back in. Okay, so you're going to get net movement out, which means that the calcium concentration in the SR lumen is going to drop further until at last the amount leaking out will be reduced down to a level where it is once again in balance with the amount being brought in. Okay, and that new balance will be achieved at a lower sarcoplasmic reticulum calcium concentration, because when you have a lower sarcoplasmic reticulum calcium concentration, that will reduce the leak back down. Okay, right, so again, this is going to contribute to the sarcoplasmic reticulum calcium luminal content being reduced again in these hypertrophied cardiomyocytes. Okay, and that then is going to mean that when you actually produce a calcium spike, because the cardiomyocyte has been stimulated, that calcium spike is going to be reduced. Okay, so both of these mechanisms, the reduction in functional circuit 2 by the absolute reduction in circuit 2 and the rise in phospholamban, and also the increased leakiness of the type 2 ranitidine receptors, these contribute to the calcium level in the sarcoplasmic reticulum being hugely depleted at equilibrium, and that means that the calcium spikes that these cardiomyocytes produce is hugely reduced, and that means that they gradually just lose their ability to contract, basically, become weaker and weaker and weaker. And you have to remember that this is happening to all the cardiomyocytes in the left and right ventricle. Okay, so the left and right ventricle are just becoming weaker and weaker and weaker, okay, because of those chronic 4 conditions, remember, that we summarized, and I'll just get them back here. Okay, these chronic 4 conditions, the hypertensive mean arterial pressure, which caused pressure overload on the cardiomyocytes, the in chronic sympathetic stimulation, which was stimulating beta-1, beta-2, and alpha-1 adrenoreceptors on the cardiomyocytes, the increased central venous pressure and pulmonary venous pressure as a result of the increased water retention, okay, and also the chronic stimulation by angiotensin II, which drive this hypertrophy, and they just basically cause the heart to try and make this adaptation to make it stronger. It buys you a little bit of time because it works for a while, but then nature hasn't quite mastered this. The cardiomyocytes work better for a while, and then for some reason their calcium signaling mechanisms just become deteriorated, basically, and that completely ruins their ability to contract and generate force. Okay, right. And that then will lead to the cardiac output gradually going down and down and down, and even with the chronic sympathetic stimulation telling the heart to beat harder and beat faster, you still can't maintain an adequate cardiac output. And when that deteriorates to the level that it's no longer sufficient to meet the metabolic needs of the body, even when you're shunting loads of your blood to the heart and brain, then it will lead to death. Okay, right. So, in the next video, what we're going to turn our attention to is the medications for heart failure. Now, most of the medications for heart failure work by blocking the neurohormonal mechanisms, by blocking this progression that is driving the hypertrophy of the cardiomyocytes, which gradually leads to this deterioration. Okay, and trying to return it back to this original state that we had where the heart just dealt with it on its own, okay, and you didn't have the neurohormonal mechanisms, okay, because this was stable. Yes, okay, the stroke volume was reduced from what it should have been, but the rate of hypertrophy was much, much less in this sort of case than if you have the neurohormonal mechanisms. Okay, so trying to stop hypertrophy and that deterioration is the main focus of heart failure medications.